Well, it's a pleasure to be here and participate in this symposium for Professor Khurana. I didn't really know him, but as Lloyd has made the case, I'm obviously his great-grandchild, because we did develop technologies for DNA synthesis with the help of automated technologies for DNA synthesis, for DNA sequencing. And actually, we developed the inkjet technology that Agilent uses now. So we've used in space the chemistry he developed. What I'd like to talk about is what I think is one of the most incredible times in all of biology and medicine, and that is how this new view of biology called systems biology is transforming how we think about medicine, how there are exponentially changing emerging technologies that are going to enable this medicine, and maybe we'll have time to talk about some of the consequences of this. So let me give you in 2 slides my view of the essence of systems biology. I've always argued that it has to be built on a platform of cross-disciplinary biology, that you have to bring together all the scientists, they have to speak one another's languages, they have to work together effectively in teams. But really important is the idea that in the end, systems biology is biology, and the driver has to be a deep desire to understand the frontier areas of biology. And in fact, it's the biology that should dictate the nature of technology you should develop, and similarly, the nature of the computational and mathematical tools that you bring to play. And I would argue good places in systems biology do all 3 of these effectively. And there are many places, obviously, that do. I think new tools are really, they're the leading edge of the transformation of biology and medicine. And we'll talk in some detail about that in just a moment. So what exactly is this thing we call systems biology? Well, one is the idea that you take all of the information that you have about a given biological system you'd like to understand, and you model it. And you can model it descriptively, graphically, or mathematically, depending on the level of information. And then you formulate hypotheses to test that model, and you use high throughput and low throughput approaches, and you integrate that data into the model, you reformulate the model, and you continue this cyclic process, and you bring experimental data into apposition with your theory. And the key thing in the end is that you create models that are fundamentally predictive. And again, they can be descriptive, they can be graphical, or they can be mathematical. And I think many people in systems biology push way ahead of time making mathematical models for everything, because the models, if they aren't embedded in real data, end up being approximations that often are not useful. Systems biology also is about global measurements. That's looking at all genes, all messages. We can't do it, but we will be able to soon, I think. All proteins, all metabolites. And the idea is we don't know, even in the simplest biological systems, all the components that participate. And I'll show you how important these global measurements are in just a moment. We have to be able to integrate together the different levels of hierarchical data, from DNA to RNA to protein to interactions all the way up to cells and so forth. And the integration of these different levels of information is critical, because the environment impinges on the digital core of information and modifies it at each of these levels. And this integration of information is operationally extracting those environmental contributions, and until you do that, you don't have a really detailed understanding of systems. And of course, information in biology is mediated by networks. These biological networks are dynamical processes, and we have to be able to visualize the dynamics across the various time points of biology. It can be evolution, it can be development, it can be physiologic responses, or what I'll talk about is disease. In systems biology, we can be discovery-driven. That is, we can say, we'll define all the elements in an object, like defining the genome that gave us all the genes, and by inference all the proteins. And it can be hypothesis-driven, and it can be done on a very large global scale or obviously on a smaller scale. And in the end, what is really going to be critical to systems biology is that these measurements are all going to be quantitative, one way or another. So the fundamental hypothesis of systems biology then is the idea that in a diseased organ, one or more of these biological networks becomes disease-perturbed genetically and or environmentally. That alters the envelope of information that network expresses, and it's this altered envelope of information that mediates the pathophysiology of disease. But it's that altered envelope that also, as I'll show you, gives us new approaches and insights into thinking about therapy and diagnosis, and I'm really going to focus on diagnosis. We spent the past 5 years thinking about a systems approach to disease, and the disease that we've looked at is prion disease in mice. This is a disease in which a normal protein has its three-dimensional structure altered, and in doing so becomes infectious in two senses. One, that infectious particle catalyzes the conversion of normal prions to their infectious form, and the disease is autocatalytic, mainly manifest in the nervous system. And number two, that infectious prion is resistant, in most cases, to hormone proteases. So when it is taken up into nerve cells, it forms the aggregates that probably lead to dilation and the symptoms of prion disease in mice in our case. So the approach we took was really a straightforward one. Let's infect mice, and then let's follow them across the onset of the infection, and let's compare the transcriptomes of the infected mice with their normal counterparts and do a subtractive analysis so we can see what the delta is, what the change is that's been induced by this whole approach. And of course, what is staggering is, as you'll see in just a few moments, you can generate enormous amounts of data. And I'll tell you, there is one, I think, utterly overwhelming problem in systems biology today that I think a lot of people aren't aware of, and that is with the global approaches we can take in genomics and proteomics and so forth. Now, the question of signal to noise is overwhelming. So if we do a genome-wide SNP analysis and you get a list of 1,000 genes whose behaviors have changed and they're ordered, you can be sure that 98% of that list is noise, and there is a very small fraction that's really going to be signal. So the really key question is, how do we deal with signal-to-noise kinds of issues? And there are two ways of doing it. There's one way which centers about really a deep understanding of the biology and being able to use subtractive analyses, and I'll show you that in a moment. And of course, you can use statistical methods that will deal more with instrument noise and things like that. So the approach that we took was not to infect one strain of animals, but to infect really 3 different inbred strains of animals, several congenics, ones that were a double knockout for the prion gene or heterozygous against a null for the prion gene or overexpressed the prion gene. And we found that by using these various types of animals and carrying out subtractive analyses, we started with a horrifying total of about 7,400 genes that changed across all of these 8 strains of mice. And we were able to reduce through these subtractive analyses down to a core of about 333 genes or so. And the subtractive analyses that use deep biology, I'm not going to go into in detail, but clearly subtracting normal against disease was really the first start, but that got you to 7,400 that changed. Then a really interesting subtraction was to subtract the double knockout against these, because the double knockout never gets a disease, so any changes it has are irrelevant. But then we could look at biological properties of the disease. For example, there are multiple strains of prions that cause the disease in different forms. We could subtract those out because they weren't central to the core. Or there are long induction and short induction period genes, and we could create strains where we subtracted those out because, again, they weren't central to the core. But anyway, we got down to a core of 333 genes. And what we did then was to map the dynamics of those genes, because we looked at them at 10 time points across the onset of infection, onto 4 major classes of biological networks that mediated the 4 features of this disease we understood from really detailed pathophysiology. I know this here: prion accumulation, activation of glial cells, and the degeneration of nerve cells, and finally the death of nerve cells. So what we did then was to integrate these 4 different types of data: our subtractive analyses, the network dynamics, the histopathology, and finally the clinical symptoms. And let me show you just a couple of panels at different time points in the onset of the disease. This is one of those networks at about 6 weeks or so. And the yellow means normal and diseased animals. Transcripts have exactly the same. The solid lines indicate subnetworks that mediate various aspects of that overall network type of disease. At 10 weeks, you can begin to see changes. Red indicates increased, green indicates decreased levels in the infected animal transcriptomes. And in this case, the first activation is that of the glial cells, which we knew from the histopathology. At 14 weeks, you can see more changes, and finally at 20 weeks, you can see the maximal changes, and the animals are exsanguinated at 21 or 22 weeks. So what we can do, not only with this network, but with the 3 other networks then, is draw the following conclusion. 2/3 of these 333 genes basically explain all of the known pathophysiologic events of this disease, mostly known again through histopathology. We have the histopathologist on the analysis team that had done all of this classical work, and he said this was the first time that these results integrated together in an organized way and very beautifully explained the histopathology. But the really important point was 1/3 of the genes gave us modules that were completely new, that people hadn't known about before, that opened up new avenues for exploration. And I would argue most diseases are just like icebergs. We know we get that upper tip, but there's an enormous core below that we'll only get with global analyses. We were able to show that 3 out of 4 of these networks really behave in a very similar fashion in several different human diseases. And what's interesting about animal models is once you can deconvolute the information to the network level, animal models can be very useful even for things they aren't direct correspondence to. And we've learned a lot about the details of what happens in, probably happens in Alzheimer's disease, and we're checking it now. The study has given us new insights into thinking about drug targets that might be really interesting for early intervention in neurodegenerative diseases. And the essence of that intervention is, can we intervene with drugs at early times at key-nodal points in the network that may return that network behavior to normal, or at least abrogate its more deleterious activities. And then, of course, it opens up really interesting new implications for systems approach to blood diagnostics, and that's really what I'd like to talk about now. So if we look at 3 different panels, we can see in this middle panel at 12 weeks, 6 weeks before there are any clinical signs whatsoever, that there are changes in the proteins. So a really interesting idea is, do these transcripts encode proteins which are secreted into the blood, and if we measure them in the blood, could the altered blood concentrations be a proxy for pre-symptomatic diagnosis of the disease? And the answer to that is yes. But there is one big reservation in using just any nodal point in the network. And that big reservation is most of those nodal points are synthesized in networks present in multiple tissues. So if they change in the blood, how do you know which tissue has affected that kind of change? So from that kind of thinking, we developed deep transcriptome analyses of now more than 40 different organ systems in mice and humans. and we've compared the individual transcript in each of those organ systems against all others to find transcripts that are organ-specific or relatively organ-specific, and there turned out to be a surprising number of these. So for the brain, we have probably 350 organ-specific transcripts. So the really key question is, are they secreted into the blood? And again, the answer turned out to be surprising, and that was that a surprisingly large number are secreted in the blood, and those secreted in the blood come from three sources. One, classically secreted. Two, membrane proteins that are cleaved by proteases, and this is a very significant fraction of membranes in the blood. And three, if you have apoptosis, you get nuclear and cytoplasmic proteins in the blood. In fact, you can measure indices of cell destruction with those kinds of proteins. So we've actually now identified 50 brain-specific and 50 liver-specific proteins, which we can assay, and I'll tell you how we're going to assay them in the blood. And of course, the really interesting idea is the following. If you'd like to measure the 50 proteins in the brain-specific blood fingerprint, we know that if the brain is normal, there'll be one level of concentrations, okay? We know that if you get glioblastoma, a few of those will change in accordance with the cognate networks that have been disease-perturbed in the brain. But if you get Alzheimer's, a different set will change. So there'll be a diagnostic ratio of changes that will be pathognomonic of each disease. And we've demonstrated this in several different disease systems in mice. So the idea is, if we could do this for each of the 50 or so organ systems present in a human, will there be a time in the future when we can take a fraction of a drop of blood, make 2,500 measurements, and assess each of the organs? And for each individual, you can make the assessment of, is it normal or is it diseased? And if it's diseased, which disease does it have? The other point that's really key is in personalized medicine, I'll be talking about in a moment, each person is their own control from the transition from health to disease, because the thermostat of protein concentrations will be set differently according to the genetic polymorphisms that you may have. So these organ-specific blood markers can be used for diagnosis. They can be used for early diagnosis, for the stratification of different types of a disease that appears at least phenotypically similar. And of course, that's important for the impedance match against therapy, for following progression and finally following therapy. And what we've done more recently in prion disease are two things. One, we've now looked at 45 of the brain-specific proteins, and we've shown at the later stage, but still preclinically, 15 of those can be changed in absolutely diagnostic ways to predict the nature of that disease. We've shown also that microRNAs can be used in a diagnostic way, and in very, very powerful diagnostic ways. And in fact, microRNAs are conserved in the blood far better than proteins are, ironically. We can use these fingerprints to assess drugs in individuals, either in personalized medicine or in clinical trials, and we're particularly interested in assessing toxicity. So we'd like to get the major organs of toxicity: liver, kidney, heart, muscle, brain, and so forth. Wellness assessment, longitudinal data gathering normal patients. I'll talk more about that. And of course, we can really do human biology by assessing what is going on in all organ systems across development and aging and various treatments. And here's an example of one of the 15 markers that we can see beautifully 8 weeks pre-symptomatic arrival and everything. So let me talk very briefly about, or list initially, and I'll talk about a couple technologies that are transformational. High throughput sequencing, it's obvious to all of us. We're using targeted mass spectrometry for discovery and validation of the blood fingerprints now. I'll say a word more about that microchip analyses of proteins. This is a collaboration with Jim Heath at Caltech. And single cell analyses, I think those are all going to be transformational, as well as the new imaging tools. Rudy Abersol has really pioneered the use of MRI mass spectrometry and triple quadrupole to be able to look at 100 proteins in an hour quantitatively, and that's the kind of assays that we're using right now. With Jim, we've developed a protein chip that has the ability to make 25 measurements using ELISA assays from 300 nanoliters of blood, dynamic range of 10 to the 6th to 7th, sensitivity of high attomole, and 5-minute turnaround time. We've already put this in the clinic at UCLA and looking at drug responses to several tumor analyses. And of course, click chemistry is a new approach to capture agents where you can use peptide 6-mers and couple them together in appropriate three-dimensional orientation so that you can create then dimers or monomers that have affinities that can increase by several orders of magnitude as you couple successively these things together. And we're hopeful that these approaches will in time be able to replace antibodies, and they'll be the key to what we use for the 2,500 organ-specific blood protein measurements in the chips. Single cell analyses, I won't talk about the technical details. I think they're really straightforward, but let me just run through some of the measurements I see doing on every single patient in 10 years. 1, I think we'll have genomes. 2, I think we'll be able to take 1,000 individual cells from something. In fact, what we're doing is taking 1,000 cells from each of 5 different glioblastomas and analyzing initially at the level of 400 transcripts quantitatively how those cells look. And the whole question in cancer of whether individual cells have quantized states or whether it's a continuous distribution, I think it's one of the most fundamental questions we'll be able to get at. But in the future, we can use that for T cells or B cells. We'll be able to make the organ-specific blood fingerprints. And of course, we can look at B and T cells and get an assessment of past history and exposure. And one of the things I'm very high about is the ability to be able to make from every single individual patient iPS cells that then can be differentiated to tissue relevant to the disease in the family and so forth. So this is going to take us to P4 medicine, predictive, personalized, preventive, and participatory. And boy, standing there looking at me, so I think I have to end. I won't go into that in any detail, but let me make one last final point, and that is, I think biology is moving toward a digitalization. That is, we'll have the ability from one molecule, one cell, one tissue to extract disease-relevant information. And I think this digitalization, together with what the systems approach to diagnosis, therapy, and ultimately prevention is going to do, is going to lead in the future to a dramatic downturn in the ever-escalating costs of healthcare. And I think that downturn could be quite dramatic, to the point where we could export P4 medicine to the developing world, and this could lead to a democratization of healthcare that really had been inconceivable before. So the question is how we can accelerate that whole process. And I think what we can use to accelerate that process, what we're doing to accelerate the process, is creating very special strategic partnerships that make available very large resources to attack these deep and fundamental problems in ways we couldn't do with conventional grant funding agencies. So we, for example, have just made a strategic agreement with the state of Luxembourg that will bring to ISB about $100 million over the next 5 years to attack what I think are the two fundamental issues in P4 medicine, namely how we translate genomes, how we translate phenomes, and integrate that data into predictive models about outcome disease. Thank you.